All right, well, welcome everyone, and thanks for joining us today. My name is Peter Washington. I'm one of the co-organizers of the UCSF AI seminar, and we're here today to celebrate Atul Butte, who, as we all know, has left a profound impact on UCSF in many, many ways, including the computational infrastructure that he has left behind at UCSF that has enabled a wide breadth and depth of high impact projects, some of which we will hear about today. So Atul has created many technical infrastructure tools that we all use: information commons, Wynton high performance compute, several NLP services, Spoke, and many others. And these tools have powered a variety of research projects here at UCSF that enabled them to really become high impact projects. So we'll hear about two today. One from Marina Sirota, who is the director of the Baker Computational Health Sciences Institute, who will be talking to us today about precision Alzheimer's research. And then we'll hear from Julian Hong, who will talk to us about precision cancer research. So without further, well, actually first, a few logistics. So the way we're gonna do this is we're gonna have both presentations go back to back and roughly 20, 25 minutes. And then after that, we'll do all the Q&A at the end. And that way we'll make sure that all the speakers have a chance to fully give their presentation. So hold your questions till the end. The speakers are welcome to revise that, but just by default. And without further ado, I'm happy to introduce Marina.
Thank you so much for having me and also for organizing this event. So we're dedicating today to Atul, and thank you, Peter, for everything that you said. I wanted to show this picture specifically. This is actually from my PhD defense with Atul. I might have photoshopped it a little bit. Now we can use AI and make it look even better. But he was a truly remarkable person, and I've had the fortune to, I was one of his first graduate students, and I've had the fortune to work with him here at UCSF for the past 10 years. And his impacted my own career tremendously. But today we're going to talk about what he has done scientifically and kind of laid the foundation for many, many different things. He actually is one of the only, I don't know if the only, but what's remarkable about his career is that he kind of pioneered 3 different fields in 3 different places where he was at. So he did his graduate training at Harvard, where he had a research line of work with Zach Kohani using gene expression data at the time and building relevance networks and developing those approaches. Then he moved to Stanford, and that's when I met him, and laid the kind of pioneered the field of translational bioinformatics, bringing different types of omics data for discovery of diagnostics and therapeutics. And then he came to UCSF and pioneered clinical data sharing and AI in clinical data sharing and analysis, impacting so many projects across the infrastructure that he's built here with Information Commons and his cross UC efforts. So I'll give you some snapshots from my own research career that kind of tie to different areas and the ways that Atul has impacted me. And I think that quote is pretty, is something Atul would say: things are only possible until they're not. And that truly motivates the type of work that we do in my lab, but also more broadly at Baker. All right, I have a snapshot of my first email to Atul. These were pretty impactful emails in my career. One was in September 2006, where I was asking him to rotate in his lab as a first-year graduate student. And then on July 1st, 2007, I asked to join his lab. It's been a while. I'll also want to mention we're doing a number of things to honor Atul, but these are 2 things that we wanted to share on behalf of the Baker staff. One is Atul Butte Innovation in Biomedical Data Science Student Award, which will start soon. And the idea is to really highlight a student who's doing innovative work, and the winner will be kind of highlighted and selected, presented at the 2026 UCSF Research AI Day. I don't know how many of you have been to our inaugural Research AI Day that took place this year. And then the second one is the Atul Butte Company competition at PMWC. PMWC is a Precision Medicine World Congress that happens every year for the past, I don't know how many years, 15 years maybe. Or so, and this will be a company competition. The conference will take place on March 6, 2026. So these are just a few things that we're doing. Just wanted to put them on your radar. All right, so now I'm gonna jump into the science aspect of things. And I always like to start by saying why I'm excited to be a computational biologist. There's a lot of data out there, whether it's molecular data, we're starting to figure out different ways to use omics, genomics and transcriptomics, but we can look at antibody repertoire, epigenomics, proteomics, microbiome, and many of these in a single cell level. And then there's clinical data, and all of these provide incredible resources to better understand health and disease and impact diagnostics and therapeutics. Of course, computational work, the type of work that we do, from statistics ranging to classical machine learning to newer generative AI approaches, provide an amazing opportunity to bring these types of data together. So in my lab, we work on bringing these different types of data, whether it's molecular or clinical data, to better understand disease. And we work a lot in the space of women's health. So pregnancy outcomes, such as preterm birth and recurrent pregnancy loss. We study autoimmune disease and also neurodegeneration. And both of those from the perspective that these diseases potentially impact women significantly more than men, and we want to understand why. And today I'll focus on our work relating to Alzheimer's disease. Many of you are probably familiar with the Information Commons resource that has been built here at UCSF over the past 10 years by Atul and the rest of the Baker Computational team in collaboration with many others. So this resource contains structured clinical data across about 8 million patients, over 135 million encounters and 40 years of activity. We have clinical notes that are de-identified for research purposes. Nearly, that number is a little bit out of date, 200 million notes. Imaging information, so over 82 million images, and some genomics data, and you might or might not hear about that in the next presentation. All the data is de-identified, accessible, and ready to be used. There was also an amazing opportunity that and kind of framework that Atul has built as part of the CDI2 group across the UC system. So these are the data sharing efforts across the state of California, which is more narrow data. It's not as deep. So about 12 years of data, 9 million patients. And in terms of genomics, cancer is the focus. And really, again, this is an amazing, this platform, whether it's Information Commons or the UCDDP, the Cross UC platform, are amazing opportunities to ask questions about disease and understand the manifestations of disease across different populations. There's also opportunities to leverage newer large language models. These are efforts led by the ARS group, specifically Keli and his team, that allow us to have a HIPAA compliant LLM on top of these data and allow to interact with it both through Versa chat as well as Versa APIs. And these tools are constantly being updated. So I wanted to give you a few examples of how we've been leveraging these data. This is work by a very talented MD PhD student in my lab. Alice has graduated already. And the first question that she was interested in asking was, how can we use real world data to better understand Alzheimer's disease? So we started with the UCSF data, identified using ICD codes, nearly 9,000 Alzheimer's patients, and identified a matched control group. And again, this is based on structured information. So diagnosis codes, lab test results, medications, everything that gets captured when somebody goes to see a doctor. So these are dimensionality reduction plots where each dot is an individual patient. This is the data at UCSF. When we started the study, we did not have access to UC-wide data yet. The other plot here on the right-hand side is data from Mount Sinai. And these are representations of the clinical profiles of these patients. And you can see that if we look at cases and controls, there's a little bit of separation, not too stark, but there's something. We can start building comorbidity networks to better understand what does the disease population look like and what does the control network look like. And you see a lot less connectivity in the controls. We can look at these separately in men and women, and we see that those are different. And we can look at specific associations. So in terms of those associations, it's not surprising that we identify things like MCI and dementia as associated with Alzheimer's. But as we go down that list, we see things like urinary tract infection, hypertension, hyperlipidemia. And on the other side, we see some associations with cancer. We can see that these associations actually hold up pretty well across the UCSF and Mount Sinai data, about 0.8 correlation, even though the patient populations are very different. We can do this type of analysis separately for men and women, and there are some differences. There are some associations that are uncommon, significant in both, and some that are sex-specific. So that was our first study. It came out a few years ago in Nature Communications. But we really wanted to see then as the next step, well, can we actually predict which patients are more likely to develop Alzheimer's? This is a follow-up study also by Alice that was published in Nature Aging in 2020. So here we intersected the UCSF electronic medical records data together with the database from the Memory and Aging Center. And that allows us to identify a really well phenotyped cohort. So when we go back and link them up, we now are very certain that these patients, we have a good Alzheimer's patient population at UCSF. And then the question that we're asking is, can we predict Alzheimer's 1 day, 1 year, 3 years, 5 years, or 7 years prior to diagnosis? And again, we have this discovery and validation system between the UCSF data and the data across the state. And we use Spoke, which I'll introduce in a minute, for interpretation of the actual features. So this is the cohort that we are dealing with. We had 5.5 million patients to start with. When we go to patients with Alzheimer's who have longitudinal data across 7 years, of course, it's significantly reduced. Nonetheless, when we match the cases to controls by a number of demographic criteria, we see that our models perform fairly well, about 0.7 accuracy for prediction of Alzheimer's, up to 7 years prior to diagnosis. All right, so now we have these models, but what are the clinical features that are driving these predictions? And we see some of those associations that we saw before, like hyperlipidemia, hyperlipidemia, memory loss, vitamin D deficiency. But we start identifying things that are a bit more interesting. We also build these models in a sex-specific manner, so for men and women separately. And we saw, again, there are certain features that are common, like hyperlipidemia, congestive heart failure, but others that are sex-specific. And there's this one that caught our attention. We found that osteoporosis was predictive of Alzheimer's in women, but not in men. So this is where we turn to the interpretation, and we partnered with Sergio Baranzini, who developed Spoke, a knowledge network. It's a database of databases that contains over 50 data sets, 50 million nodes across 26 different types. So genes, proteins, drugs, and many, many connections. We wanted to layer that on top of our clinical features. So I'll zoom in here. These pie charts represent, well, here, they represent the features that are consistent across all those time models. And we've kind of zeroed in on two: hyperlipidemia and osteoporosis. When we look at the spoke, so in the middle, these are the spoke nodes that connect these clinical features. We saw that the links between hyperlipidemia and Alzheimer's, we saw ApoE as one of the strongest links. Not too surprising, that's the biggest risk factor for Alzheimer's in terms of genetics. And of course, also involved in lipids related pathways. But when we look at osteoporosis and Alzheimer's, we saw all of these immune genes coming that we did not necessarily know about before. Then we were further able to validate just the clinical association for both hyperlipidemia and osteoporosis across the UC system. You can see that for hyperlipidemia, when we stratify into males and females, the relationship exists in both. When we look at osteoporosis, it only exists in females. So final layer of data here, we wanted to see if we can use genetics information from entirely independent groups to link the relationship between heel bone mineral density and Alzheimer's. So this is showing results of GWAS studies, and this was done again in collaboration with Sergio and his team, split into females and males. And you can see that in women, there's this peak in the top right corner that we don't see in males at all. Same thing by looking at expression through EQ-tails. You can see a peak here when you look at everybody, but when you stratify, it only appears in females. So it turns out this peak is around this MS4A6A gene, which is an immune gene involved in a number of different pathways through inflammation. This type of analysis doesn't tell us what is going on mechanistically, but it tells us that maybe there's something in the relationship between osteoporosis and Alzheimer's, specifically in women, that might be mediated by this specific gene that needs to be explored further. So there's a number of projects going on in the lab now following up on this work. There's a preprint by Alice in collaboration with Pedro Pinheiro Chagas, where we're incorporating clinical notes leveraging large language models to be able to predict not only Alzheimer's, but also other related dementia phenotypes. So integrating both structured and unstructured data. And we're seeing, this is very preliminary, but quite some improvement on our original models that I presented. In the lab, we've also been looking at social determinants of health in the context of Alzheimer's. We've been doing some unsupervised clustering to identify subtypes of the disease, as well as looking at the links between Alzheimer's and other conditions like autoimmunity and also cardiovascular disease. So I've really shown you how we can use different types of data and how the groundwork of organizing and making these clinical data sets available can allow us to not only predict which patients are more likely to develop Alzheimer's, but also to understand why and what might be some of those relationships. Of course, the models need to be further validated, and we need to just explore the biology and maybe potential therapeutic potential of MS4A6A specifically. How much time do I have? 5 minutes? Okay. So in the last 2 minutes, I wanted to share another study that recently came out from our team, switching kind of more to the therapeutic side of things. This is around drug repurposing, and specifically cell type specific drug repurposing for Alzheimer's disease. This is work in collaboration with Yidong Huang that was led by Yachao Li, who was a graduate student in the PSPG program here in the lab. And this work really stems from my own graduate work with Atul. So the hypothesis behind computational drug repurposing approaches that we've applied is that we can use gene expression to identify drugs that potentially reverse gene expression signatures of disease and could help patients. So this was the original paper from my graduate work published in 2011, both the methods and the application to IBD. Since then, we've applied it to other autoimmune diseases like dermatomyositis, but also cancer applications. we've explored it in the context of COVID-19. But today I'd like to just touch on the approach of using this method to identify cell type specific combination therapy for Alzheimer's. We start out by looking at data. So Yachao aggregated 3 different data sets and developed cell type specific signatures for Alzheimer's that she then queried against a resource called the connectivity map, which is cancer cell lines treated with different compounds, looking at reversal of that expression. Through this analysis, she was able to identify a number of drugs that modulate several cell types. So we started with 1,300 compounds, 85 candidates across different cell types, 85 candidates that reversed expression at all, and 25 across different cell types. She then leveraged electronic medical records, same data I've been talking about, across the UC system to see, since these are FDA-approved drugs, which drugs, patients on which drugs have a lower prevalence of Alzheimer's. And there were 2 cancer drugs that floated up to the top of the list, letrozole and irinotecan. So now we were able to funnel from 1,300 to 2 that we ended up testing in animal models. Letrozole is an aromatase inhibitor that blocks the conversion of androgens into estrogens and used for breast cancer. And irinotecan interferes with DNA replication and used as a chemotherapy agent. So there was an extensive animal validation studies of this work where we used a cross of 2 pretty aggressive Alzheimer's models, used a combination of the drug, each drug alone, and untreated. I'm not an animal model expert by any means, but this is a Morris water maze test where the animals are placed into water. As I understand, they don't like that. They try to get out. One of the quadrants has a hidden platform that allows them to get out. And what we're looking at is the time that it takes to escape. And you can see that both at the 24 and the 72-hour probe, the combination therapy allows them to do that more efficiently. Yachao also measured a number of different pathology markers, including hippocampal volume, amyloid beta, tau measure, looked at microglia as well as astrocyte markers, as well as neuronal staining. And these are all summarized in this table. And you can see that some of them are significant with individual treatment, but they're all do better in combination. And then as the last step, we looked at actual samples from those treated animals to see if the human data that we used for the discovery of these compounds are actually replicated in the animals after treatment. And we saw significant reversal of, well, relevant pathways, but also the specific human genes across the data from the animals. So you can see the Alzheimer's signature here that's listed first, and then combination therapy treatment, and you can see reversal across all of it. So again, this is another example where we now start with the molecular data, gene expression data, generate a hypothesis, use clinical data as another layer to validate it, and then finally end up using partnering and seeing what these drugs do in animal models. We're hoping that this specific combination could go into clinical trials, and we're also exploring this approach for other diseases, as well as incorporating other data sets. So with that, I want to end with saying that there's a huge opportunity, and the basis that Atul has built, both in terms of the data sets here available here, the clinical data sets, we're working on bringing molecular data into it, as well as the methodology to ask new questions, whether diagnostic or therapeutics. And really, there's an opportunity to bring AI and AI-driven discovery across many scales, whether we're looking at DNA, molecules, cells, tissues, or population level analysis. With that, I'd like to thank all the people doing the work. This is our team in my lab, and I would love to take any questions at the end. Sorry.
can like barely see, by the way.
Yeah.
Well, maybe you can take questions, given that.
Okay. All right, sounds good. I can take a question. I'm so used to saying that at the end that. Okay.
So you had a strategy where you found that certain gene expression was associated with the disease, and you looked for existing drugs that could counteract that. If you take like all of the gene expression, what fraction of them do you think we're going to be able to cover with drugs that we have versus how many are we going to need to make something new?
Oh, that's a great question. I mean, I would say, so the data set that we've been using now for, I don't know, nearly 15 years, it's fairly old. It's a microarray data set, which has its own limitations. So we measured 20,000 genes, and we see that maybe, I don't know, I'll wave my hands and say maybe 70% of them are affected by these drugs, but they're also measured in cancer cell lines, which is less than ideal. What I would love to have ideally is measures of gene expression of these drugs on the relevant disease tissue. And there are groups that are moving in that direction. And there are some efforts to do that on a single cell level. So I don't know the exact answer to your question, but I think we need more exact measurements to be able to answer that comprehensively.
All right, thanks for everybody bearing with that little minor technical snafu. I can barely, just before I start, I can barely see, and I can also barely hear. So I'm going to do my best. I should point out, Angela and I were just talking about this morning, that I do not get Star Wars confused with Star Trek. But for one, I had this at home, but you know, seeing as we're here to honor Atul, this also seemed very appropriate. You know, the last time I saw Atul, I think was earlier in this calendar year, and the last conversation we actually had was about Andor. So I thought that wearing some Star Wars paraphernalia would be appropriate. So so my talk is actually titled after a quote from Atul. And I went and dug it out. Actually, he'd probably be proud. I used GPT to help me compile lists of quotes that he had given that were relevant. And really, I think, as a practicing radiation oncologist and clinician, thinking about, driving medical and clinical care with data is really, I think, fundamentally sort of what my group hopes to do, and I think really a part of Atul's vision. Though he did so much. So I've people who have seen my talks know that I like to use this like image for my little like navigation slide. What people don't know maybe is that I actually used this same image at my job talk when I was getting recruited here. And Atul came to that talk and actually tweeted the image out. So we'll just jam that in there. But we'll start by talking about sort of how I came to be at UCSF. And so like Atul, I was at the other place in the Bay Area, and then eventually made my way over here. And actually, the first time I crossed paths with Atul was actually in one of his courses. And I actually had to go back. I went to my transcripts. They don't say who the professor was, but like I had to go and dig. Then I pulled up like the Stanford course catalog, and actually I found this like very flattering photo of Atul, as well as the course description. My wife looks fondly back at that course because they had great food. So she always brings that up. I don't know if we ever got a chance to share that with Atul though. And then, so I did my residency at Duke, and one of my or my primary research mentor was Jesse Tenenbaum, who many of you may know. Jesse and Marina fight over who was.
They don't fight.
Okay, they don't fight. They don't fight. There are technicalities. There are technicalities with regards, and I can also barely hear, but I can hear Marina. But there are technicalities with regards to who is Atul's first grad student. I think Marina is Atul's first complete grad student, and Jesse
graduated.
was the first one that graduated, and so Atul also tweeted that I've got a whole collection of Atul tweets, but when I came out to interview, Atul had posted this, so I think really honored to be part of this program. So a little bit about just me and my research program. We really focus on trying to bring informatics and digital health to the clinic. As I mentioned earlier, I'm a radiation oncologist, so much of that is related to how we deliver cancer care. So we really work on trying to improve our clinical decision-making, or data-driven medicine, as Atul would have said. Work on natural language processing, wearable devices, then try to bring that into clinical trials where we actually evaluate how they alter care and how they alter outcomes, and then try to do better in our clinics. And that can look a number of different ways. The clear one I think that's really important to us is to better take care of patients. But we also, as many of us know, through a number of informatics related efforts, work on trying to improve the care experience. And I think every clinician's looked like this guy in the bottom corner. I know I have, probably on a daily basis. So Gene knows that. All right, so now with all that dawdling, we'll get into some research. So the first part is really focused on unlocking clinical data. And when I got here, it's funny, is that I think Taylor Swift is in everybody's mind too much these days, but I was thinking of different eras for Atul. And so, when I first got here, he was sort of in a real world evidence kick. So that was a big part of when I first got here. And this actually is my first paper with Atul, as a commentary that we wrote together on real world evidence and how to think about real world data. And had a chance to be part of this group that he and Vivek had put together. You can see the Marina's on the slide too. And really a lot of this was empowered by information commons. And you heard Peter mention it, you heard Marina talk about it. And it really has facilitated a huge amount of research. And I only have so much time, but I thought I'd just start spamming stuff out to people. But it's enabled many studies. And I try to, Atul is always great about shouting out mentees and supporting mentees. So just a handful of our mentees that have presented work that has utilized information commons for a lot of different types of research projects that have spanned a lot of different domains. And I thought I'd just kind of raise one paper. This is actually my first paper, I think, that our team published using information commons. So Will Chen is actually now a faculty member, so it's been a while. And so Will had been interested in, he had seen some poor mental health outcomes for some of our patients that had come through our cancer clinics, and was interested in understanding something that is really hard to evaluate in a clinical trial. That's certainly one important domain that we can leverage, real world data. And Will did some work demonstrating associations between mental health diagnoses, short-term morbidity and mortality, as well as discrepancies in how frequently people were diagnosed by race. So this is one of our early studies, and we actually, I didn't include this in the slides, but I remembered as Marina was talking about the UCY data, but in another another major contribution of Atul's to the system. But we actually have a study that's currently under review, trying to reproduce these findings across other institutions in the UC system. Then I kind of thought about the COVID era, and sometimes it feels so far away, but was not that long ago. But really, Atul, empowered a lot of very important data at a time when, we were looking for data in the pandemic. And certainly, I think a lot of people were really thankful for the opportunity to look at COVID-19 data. A number of national efforts that focused on, cancer, patients with cancer, and the implications of COVID during people's cancer therapies. And this was just kind of one study that we had put together across several that was led by Hala Borno, who's a medical oncologist here, and Eric Small as well, who's a leader in the Cancer Center. Sasha Yousefi, actually, just a shout out, was an undergrad in my lab that helped us put this analysis together. But what we were able to do is quantify some of the relationships between cancer diagnoses, other comorbidities, and COVID-19 diagnoses with mortality during the course of the pandemic. So I think this is like a popular Atul quote that's cited pretty frequently. I was able to find it on Twitter. Apparently I liked it however many years ago it was. I later found like a version of it. See, GPT really helped me out here, did me a solid. Found a version of it in an interview that he did. And one of the highlights that he said was that, you know, data doesn't do anything unless there are teams that are ready to act on that data. And that's a fundamental underpinning of what we try to do on my team. And so I thought I'd mention some data from my group that sort of feeds into things that we're doing here at UCSF. But years ago, before I had gotten here, we had created a machine learning model when I was a resident, what seems forever ago, to identify patients who are at high risk for emergency visits or hospitalizations during the course of their cancer therapy. And we later took that model and operationalized it in a randomized control trial. And at the time, and I think still, one of the few sort of implemented cases of machine learning sort of altering our clinical decision making, what we found was we could use the model to accurately direct increase clinical surveillance and clinical care, and actually reduce, we cut in half the rate of emergency visits, hospitalizations, as well as the costs, which is in this slide, which I stuck in later, so I forgot that it was in here. But one of the important efforts that we have is to try to bring this model, which the study was called SHIELD, to UCSF, as well as to other places. We've got an awesome team, and Mari sort of led the efforts here at UCSF. We're calling this Vision because I'm a sucker for Marvel references. But since Jean invited me, I thought I'd give her a shout out during this talk. And so assuming this animation works, so as an example of like trying to transfer models from one place to another, we took a look at the Duke experience. The model's positive predictive value for an ER visitor hospitalization is 20%. And so when we brought it to data here at UCSF, again, empowered by Atul's work in information commons, the positive predictive value was much lower. There's a lot of nuance to that. And so she's the brains behind the operation, but really trying to build out ways where we can understand how data is different from one cohort to another. And so taking a look at the covariates, we're able to identify places that are different, and then tailor models such that we can provide a performance boost and get things back realigned. All right, I think I probably got to pick up the pace. So, all right, so I think Marina mentioned natural language and clinical notes, a big effort of Atul's. It started off when I got here as cTAKES. Incidentally, I had done some work on cTAKES with Jesse before I had gotten here, specifically looking at treatment toxicities during cancer therapies. And so this research really just kind of dovetailed together. We were able to leverage this huge swath of data here at UC and build a number of studies around this. One of these, kind of the most recently that was published, was some work that was led by Chichi, who is a graduate student in my lab. And she was able to take sort of a huge expansive look at the sort of acute care experience here at UCSF, try to characterize the patterns of symptomology and how different patients experience things as they undergo treatment. And of course, Atul was excited about large language models. We all are, and so that was sort of, I guess, the next era that I hit. And this was work that we actually got a chance to partner on. It's led by Li Qing, who was in one of Atul's graduate students, sort of shared between me, Travis, and Atul at this point. They did some great work taking a look at, looking at large language models and how well it can retrieve information. This is early on in kind of the, I guess, like AI hype cycle, and really tried to quantify how we can get information out of very minimal additional training or fine tuning to pull information out of the chart. And sort of analyze how across different models, this can vary in different clinical scenarios, to try to give us some context for where we can, apply these models and where they need more work. And this has led to a study actually that just got accepted to ML for Health. Li Ching put together sort of what we're calling a playbook, or essentially a strategy for how we can, adapt large language models to trying to extract symptoms, really powered by a number of clinicians who contributed to some of the manual annotation. Yuta's in the room, so he really helped out with that. One of the, I think, exciting things was, applying multiple models in a cross-validated strategy to try to identify cases where the clinician annotators may be a little bit more inaccurate. So Travis had a good time sort of finding places where he needed to make revision. So it's a neat part of the study. So I'll sort of end with kind of continuing Atul's legacy. Some of you may have heard about the Wild Cancer Hub West, which is a collaborative effort between us and Stanford. It's so very appropriate for Atul to have been leading that. And I think this, this team, we're really trying to carry on his work. And really this study is really geared towards trying to answer the multiple questions that arise in the course of clinical care for a patient with cancer. And so what we're trying to do is, assemble and really extract meaning out of the data that occurs over a long-term cancer journey. And so the end goal is to create a foundation model that will enable us to better explore this cancer data, help us give better insights, hopefully be tailored towards specific uses. And with that, I'll wind down and say thank you. And I guess we will take questions now. Questions in the
room. Thank you. What do you mean by he made revisions? Travis made revisions.
Oh, yeah. So we basically, Travis, I think, did a number of the annotations during the course of the study, and we employed these models, and they basically created a vote system. And so there were certain scenarios where, like, most of the models agreed that Travis may have been incorrect. And so then Travis went back and took a look at them, and he was like, oh yeah, the models are actually right. Clinical annotation is super hard. And I find it, it's a very time consuming task, and annotators are very variable. We actually had a study years back that sort of compared annotation, and, just being able to say that someone is short of breath, like, people can disagree just based on language. So it's really interesting to see when, like, models converge on a decision. Sometimes, like, there's a lot of variability and the models disagree, the person disagrees, it's hard. But there are certain scenarios where they can agree, and then you go back and look and take and review. And I think in general, Travis was pretty impressed by the areas that they identified.
What you can hear. Anyway, a cancer journey you just showed at the end. Quick question: do you mean to tackle that all with large language models, or more than that?
Yeah, that's a good question. So more than that, but large language models are, of course, like, part of it. There's I didn't go into a whole lot of detail, but the project has different subparts. There's sort of the first part is kind of an information retrieval subpart, which is probably, I'd say, a little bit more large language model centric. And then there's sort of knowledge exploration, which is kind of more of an agent type situation. And then the third is really constructing the foundation model. So yeah, language model plays a role, kind of different pieces. And, a lot of the AI agents these days are mostly language models. So
So I unfortunately didn't ever get to meet Atul, because I'm a first year this year, but I just would have wanted to know like his thoughts, and I guess through y'all, your thoughts on there's lots of AI, there's lots of, you know, machine learning tools, lots of LLMs, like but often I think we get so caught up in like this expansive array of tools, like we don't always know where to start, or like how to start small and then grow on that. So what was his opinions on that, or like how to approach these problems or thoughts, and then maybe y'all's as well? Go on, start.
I don't know. I'm gonna try. Well, from my experience as being a graduate student with him, he was not that much about the methods. He was more about the question. What question can you ask with whatever data, with whatever methods exist, and how can you get to that answer first? If the methods are not necessarily there to answer that question, that's when innovation on the method side was. But he was an early adopter of all technologies. I mean, I remember chatting with him about ChatGPT and like very early on. I mean, he was the reason why I tried it for the first time. We were having dinner actually here at Mission Rock, and I came back from that. I was like, okay, I got to try this. Like, I've never tried. I got to try this. So he was an early adopter, and his work was very often or almost all the time driven by the question and by the problem.
yeah, I don't think I have too much to add to that. But what I'd say is, you know, when I went digging for the quotes, I forget one of the text blocks that I had in the talk. He talked a little bit about that, which was really like it's about you know, in this context, it was about solving problems in clinical care, but it was that like you know, you don't have to necessarily, it doesn't have to be like the fanciest thing. And certainly many of us in the room know that we have many problems that don't require modern solutions, so to speak. And so I think that was certainly an anchor for them. That said, like Marina said, he's definitely an early adopter of stuff, and you know, Atul get like really excited about things, you know, so so like new stuff like definitely got him excited, but I think at the end of the day it was really about solving the problem. Yeah, just I don't know, he'd just be super excited about cool stuff.
One thing I can add to that, I knew him well, is yes, he was very much the early adoption stuff. That got him very excited, but he always insisted that you clinically validate whatever you see. To him, validation was a holy word. If you're just seeing these things come out of any model, how nice is that? Won't be so nice, right? So validate or start a company.
Validate and then start a company.
Go the other way.
That's true.
Hello, I'm also a first year PSPG student, and as we're learning about all of these like large data sets to be able to predict certain kinds of diseases like Alzheimer's disease, I always wonder like how we bring it back to the patient, like how it applies directly to the individual, because like I'm imagining like a case that like a patient has a high predictive risk for Alzheimer's disease, but that could be like the mechanism isn't clear there. So how do you connect like the big model, what the big model is telling you, to like how you go to treat the patient?
Absolutely. Oh, you raise a very important question. What is the actionability of knowing that somebody has high risk of Alzheimer's? Because that in itself might be daunting, right? If there's nothing really to do. So in my opinion, the discovery, the prediction and therapeutic discovery go hand in hand. So you can imagine, yeah, we have these two, well, let's say these two cancer drugs or something else, some intervention. It could be a non-therapeutic intervention too. If you know that somebody is high risk for X, then maybe there's some way to intervene earlier. The other way this data could be useful is potentially for enrollment in clinical trials. So those patients might be more likely to be considered for a clinical trial if there is some sort of a that the computation can, the data-driven computation can lead to something.
Yeah, I think actionability is like a huge part of this. And go back to the Atul quote. You know, we've seen a number of different studies. Well, certainly a number of models that we see that get published. They never see a patient, right? But there are also some efforts that should be applauded where, like, you know, the model gets implemented, but then like the physicians aren't really sure what to do with the information. That's actually come up in some like DeepMind's work. And the actionability it becomes important. So I sort of I'll take a step back, and I sort of think about these problems like a couple of different ways. Like there's sort of the prediction problems where you're like okay with like associations, you know, having some impact that may not be causal impact in the predictions. And then there's sort of more like exploration where you're trying to do more causal inference, figure out like what the root of the issue is. And of course, I think as I frame it that way, like the prediction problems are maybe a little bit easier, but you do want to have like, you know, I think you want to always have the clinical objective in mind and then work backwards from there. So it's like, we want to do X, you know, so we need to find Y, and to get to that point, we need to do Z. There was actually, UPenn actually did a lot of like early work in the, gosh, I think in the late 20-teens, early 2020s, with various like machine learning models. And one of they used to do that thing where you like fill in the words. Sorry, I can't hear. I shouldn't have asked a question when I couldn't hear. Oh wow, this microphone's a lot louder than stuff. Mad Libs, yes, yeah, so they used to do a Mad Lib thing. So like it was like, you know, we have this problem. This is what we're going to do to reduce the risk. It's funny, actually. I went to a talk and they had said like acute care during cancer therapy was one of those problems, but they didn't have an interventional strategy, and so they basically like shelved that one. So it's kind of interesting because I heard about it after. I think I actually heard about this like a day that I was presenting like the Shield results and stuff. But I think the actionability, especially for clinicians, is super important because predictions, you know, we struggle to figure out like what to do with them if we don't have like some kind of, you know, thing that we can intervene with that's set up.
I also say for the trainees, like I'm not a so I've learned to partner with domain experts in those. Will allow us to figure out how do we use these data models, whatever we create, and what is actually useful. So I think that's really so
Yes. So what strategies can be implemented to maximize the benefits of applying AI in healthcare while mitigating potential risks, e.g. bias, data misuse?
that's a very broad question.
Yeah, I guess the fact, well, I was gonna say like the fact that the person asked the question, I think that's probably the first step, which is to be cognizant of those problems. And then, you know, I think there are best practices across like kind of each of those concerns. But you know, it's a pretty like laundry list of things that you really have to think about, I think. And you know, I don't know, I give a version of a talk that's like AI gone wrong, and it's like, you know, you can fill up like a couple hours of like different types of things that, you know, that can go wrong and stuff. And I don't know, I think the big thing is doing your best and to have a good systematic way of thinking about things.
I agree. Yeah, I think it's, you know, being cognizant of the data that goes into your models. You can either see me or hear me. If I stand here, you can't see me, or at least half of you. Okay, sounds good. I will look up. So yeah, it's, you know, the data that goes into the models, because if the models are trained on biased data, then of course it will perpetuate bias as we move forward. So these are just all considerations that we think about as we design our studies, and we do the best we can.
Okay, well, we're out of time, but let's give another round of applause to our speakers. Thanks everyone for coming, and happy Halloween.
First,
still reflect the.